Hello, everyone. It's midday here. It's probably afternoon or very early in the morning in other parts of the world where you listen to me. When Ralf approached me about this workshop and asked me to give a talk, I thought about giving a talk about how to describe a species. But that has elegantly covered by Roberto Reyes yesterday and by other people in the symposium. So I'm glad I decided against that and decided to rather give an unusual talk, which looks at these social and sociological patterns that you encounter when you become a fish taxonomist. So it's going to be a very personal talk, but it's not about me. It's about how someone like me became a fish taxonomist. I usually start my talks with these three slides I'm going to show now, just to remind you that fishes have an incredible diversity, the most diverse groups among vertebrates, more than 35,000 described species. Other people have said that 300 to 400 new species discovered and described every year. But fishes also have some of the largest vertebrates. And this is just an oarfish, a grouper, and an ocean sunfish, massive, massive animals. But they also have some of the smallest vertebrates. And these are just three species or three genera compared to a 5p coin for comparison. So it can be huge, they can be tiny, and that's the interesting part about fishes. Now, my interest in fishes started when I was 7 years old, because that's when I got my first fish tank, and that's little Ralf up there on the upper left. That is not my fish tank. Mine was much, much smaller with fewer species in it. And I was just amazed by the diversity that you could see in the aquarium shops. And one of the books that actually piqued my interest in diversity in fishes was this aquarium atlas, which contained hundreds and hundreds beautifully illustrated with color photos of fishes. And this is just a page that deals with the Trichogastus species in India. Now, when you look at my academic career, and I don't need to repeat that because Rajat gave you an introduction, it looks like a straightforward highway from 7 years old getting a fish tank to becoming a fish taxonomist, but nothing could be further from the truth. And I'm going to give you some slight insight into how I became a fish taxonomist. Now, my childhood interest in animals, in diversity, brought me to study biology. And I did that at the university in Tübingen from 1988 to 1992. But when I started biology, I wanted to become a geneticist. That was very clear. It was exciting, interesting what geneticists found out at that time. And then I took my first course in genetics and completely abandoned the idea because I knew this was nothing for me, just having little tubes with substances and you can't really see what you're dealing with. That was not me. I needed the animal that I could touch and look at, turn around. I'm now going to talk about the main decisive events in my life that actually changed the course. And why I also didn't become a geneticist is this course. This was commonplace in Tübingen at that time. Every student would have to take what is called a Großpraktikum, which means you spend an entire semester every day, 8 to 10 hours, just doing one thing. This could be botany, botanical studies, it could be physiology, it could be genetics, anything you can think of in biology. But there was also a course on vertebrates and vertebrate anatomy, and that's the course I took. And that's me with a colleague looking at a skinned fox and looking at various aspects and drawing them. But we also dissected other animals. That's an antelope, and we needed all hands on deck to actually skin this animal. But looking at these animals in comparison gave you a really good appreciation of the internal diversity. And that's a manatee that was dissected by the person on the right. He was an assistant professor, and he explained to us some anatomical features there. So I did change my idea of becoming a geneticist completely, and it's very clear that I wanted to be a zoologist and doing classical zoology. And the one person that actually changed my entire view on zoology, on systematics, on taxonomy is the person on the upper left, Gerhard Mikolait. He taught 2 very, very intense courses. And he also helped me develop my critical mind, if you will, being really critical, challenging ideas. He would always ask questions in his course. And if you give an answer, he would then say, well, why? What's your evidence? Why do you think that? And he was not just satisfied with textbook knowledge. And he also piqued my interest in entomology, and that's me collecting insects, specifically beetles, actually, during that time. So Mikolait is also still alive, and he was a contemporary of Willy Hennig, who came up with the idea of phylogenetic systematics. So that's another area where he introduced me, phylogenetic systematics and phylogenetic thinking. So he, to me, is one of the giants, one of my most important mentors. But you see, I'm still far away from taxonomy here. Now, another important individual is Melanie Stiasny, who I met during a conference, the European Ichthyological Conference in The Hague in 1991. I was a very shy student. I didn't present. I was actually paying my way on my own funds. And there I was among all these giants in ichthyology. And Melanie gave a talk, and only after her talk, I actually dared talk to her and kept in touch with her, kept asking her for advice. And she then eventually told me about the collection study grant of the American Museum of Natural History. So I applied, got the grant, which enabled me to go to New York for a month. But you need to keep in mind that I was born in a little village of just 2,000 people. Tübingen, where I studied, had 75,000 inhabitants. So going to a place like New York across the Atlantic, which actually was my first ever international flight, just scared the hell out of me. But when I arrived, I somehow managed, I had to overcome all these challenges, and I had a great time. And Melanie, in those 4 weeks, told me if I was interested in anatomy and development, I have to meet Dave Johnson. So I took a train, went down to Washington, D.C., spent a couple of hours with Dave, and came back. And if I were to give a talk on morphology, one of the main figures, the big important figures in that area of my research, my morphological mentor, if you will, is Dave Johnson. Well, Melanie then also told me about the postdoc program at the American Museum. And I did apply, and to my surprise, was successful, secured a fellowship, a postdoc fellowship. And I went there in 1995 and spent an entire year at the American Museum. And that's me just shortly after I arrived. And of course, the American Museum is also a hotspot of ichthyologists. So at some point, it was unavoidable that I ran into Gary Nelson. And he is the one who shaped mainly my ideas on biogeography. So again, another person who influenced me in my thinking quite significantly. New York was a great time. You might know that the AMNH is just opposite Central Park. So during lunch breaks, we would just get our rollerblades and go for a little rollerblading in the park. It was also during my postdoc time in New York that I did my first field trip. Melanie had asked me, there's funds, where would you like to go? And I said, Myanmar, for sure. And the reason why was because I wanted to collect these 2 fishes at the lower right. That's Paraspherictes oscillatus, a labyrinth fish, and Anabantuate, and Endostomus paradoxus, whose phylogenetic position is still being debated. I wanted to see these 2 animals. So I went to Myanmar, but the options to move in the country were extremely limited. So we spent a lot of time in the capital waiting for permits. And during that time, I ran by chance into U Tin Win. And U Tin Win was a chemistry professor at the University of Yangon, but he had a keen interest in ornamental fishes, especially local Myanmar fishes. And he had already a pretty good idea about the diversity. So we immediately hit it off, spent hours talking, and he told me that if I ever wanted to come back and travel around the country when it was possible to go to these more remote places, he would be welcome. He would be happy to help me and welcome me. So that was a very important contact. But also on my way to Myanmar, and on my way back, I stopped over in Singapore. And that's where I met these chaps. Peter Ng was extremely welcoming, extremely supportive. And so was Yokhui and Kelvin, the collections manager, and Yokhui, a fish taxonomist there. And this relationship that we developed in these couple of hours that we spent together lasts until today. And we are good friends and great colleagues. And we've done collaborative work together ever since. Now, after my postdoc in New York, I came back to Tübingen and as an assistant professor. And that's actually me just arriving from New York and in my lab office space at that time. And again, it was kind of a chance encounter. I was put in touch by a friend of mine, was put in touch with Maurice Cottelat. And Jörg Freyhoff has already told you that Maurice was one of his main mentors. And so was Maurice my main mentor when it comes to taxonomy. In the aquarium trade, I had found this little pufferfish on the left. And this had come in with the pufferfish on the right, which is Carinotetradon travancoricus, a known species. And it was pretty clear that there were differences in color pattern, differences in the sexual dimorphism, but most significantly, differences in the arrangement of the spines and their shape. So it was pretty clear to us, this is a new species. And I contacted Maurice for advice, and he also had topotypic material of Travancoricus for comparison. So it was clear to us that's a new species. We just didn't know where it came from, aquarium trade, right? So we held off with the description, tried to find out more about where it may occur, and actually contacted colleagues in India, and nobody had seen this fish. Actually, some doubted that it is a species of its own. So after waiting without any success, we decided we're going to go ahead, describe it as a new species, and maybe that will trigger some people to go into the field and find out where its actual type locality is. And that never happened, but I'll get to that later in my talk. Nobody ever found this species anywhere in the wild. Again, it was a chance encounter when I went to an international symposium in Porto Alegre. That's actually where I met Roberto Reis the first time. And that's also where I met Sven Kuhlander from the Swedish Museum of Natural History. And he had plans to go to Myanmar. And he had started to work on those groups that I was interested in. So he immediately offered to collaborate with me. And again, Sven is a very important person for me because he introduced me to Fieldwork. And he invited me in 1998 to go to Myanmar to collect with him. And that's what we did. I joined his collecting trip. And one of the goals was to go to Lake Indoji. That's where these two fish on the lower end of the slide occur. And we made it there. We collected these fish. So one of my dreams of a few years before had come true. And Sven also showed me the ins and outs of collecting. What kind of nets? How do you preserve? What do you use? Where do you catch fish? And so we were a great team by the end of the trip. And some of that material we collected led to our revision of the Badidae. And that has been a topic in Lucas's talk. I don't need to repeat that, but we were able to describe quite some diversity that was unrecognized before. Now, after my time as assistant professor, Dave Johnson, who, remember, I had met on this chance trip to Washington because Melanie had recommended it, was keen to develop a proposal with me to invite me as a visiting scientist to the Smithsonian. We submitted, the proposal was successful, and he had money to support me for 2 years. So I went there, and I would still consider the Smithsonian the hot spot, the best place you can be if you are a systematic ichthyologist, taxonomist, because there were several really, really big names in systematic ichthyology, and I was able to meet them, talk to them, and even eat a durian with them in the lower left. That's Bruce Collette and Lynn Parenti. I had an absolutely fabulous time there in these 2 years. But again, there were also funds to do field work. And I went back to Myanmar in 2003 and collected, now with a much better idea of what to do, and came back with quite a substantial collection. And eventually, because during those 2 years, it just happened that a position opened up at the Natural History Museum. I applied, and Dave was one of the people who wrote me a glowing reference, and I interviewed and got the job eventually. So in 2004, I moved to London and I joined the Fish Group, and even became, for a little while, the Natural History Museum's poster child, as you can see, poster boy on the lower right. For various reasons, I can't go into any details here, I left the museum in 2019 and joined the place where I'm at now. And you can see my lab office space in the background. That's the Museum for Zoology of the Senckenberg Natural History Collections in Dresden. That is a rough outline of my academic career, with all the little chance encounters and points in time when I could have gone into a completely different direction had it not been for a number of things to fall into place for me. So far, and that's only my taxonomic work, my morphological work would actually fill another separate talk. That's the taxonomic work I've described, as Ralf said, 72 species, 7 new genera, and even 2 families. And these are some of the fishes that I co-authored or described in the last couple of years. Now, Myanmar has been my main center of interest, and I've actually gone there 10 times. I counted them up recently. 10 times I've been to Myanmar over a period of almost 20 years. And I've been to many, many different remote places, and I've seen how the country has changed in those years, but it's still a wonderful place to visit and do field work in. And crucial, instrumental in planning every single trip has been U Tin Win and his family, his wife and his son. And when U Tin Win passed away, unfortunately, way too early in 2014, it was his son that helped me organize the other more recent trips. But without their help, I wouldn't be where I am. I would not have the Myanmar material that I have been able to work on. And as Rameshwari said, fieldwork can be quite tough. You know, we collect in different habitats that are a very muddy pool that is just initially quite difficult to get into, but once you're in and when you drag your net, get your first fish, you're excited and you continue. That's a pristine river at the lower right near the Himalayas, and this was in Putao. This is along the Tenasa Rim range, and there we fish with little, much smaller dip nets with a very fine mesh to get certain species of fish. And sometimes you have to really work hard to get the fishes that hide in between the rocks. And this again was a kind of a special habitat because a very, very cold stream in the very far north of Myanmar, where we got things like Diptothorax, Ampliceps, or Lyra, they would occur there. And of course, whenever we go and to these different locations, we try to take coordinates, of course, that's standard, but also some water parameters. And most importantly, I would say we try to photograph fish alive in a photo tank. And when you're in a remote place, you can actually concentrate, focus in the true sense of the word on getting a good picture of the fish. But sometimes you just attract attention. And if you have lots of people around you who just stare at what you're doing, it's quite difficult to focus. But still, you need to learn how to deal with these situations. And what you get then, if you have a decent camera, and if you're lucky the fish doesn't swim around too rapidly, you get a good shot of the live coloration. And if you have a higher resolution camera, you can zoom in and actually see lots of details. So that's quite important for those fish species I work with. And I would argue for most fish species, actually, it's important to get a live color image. Then we preserve the fish in these Nalgene bottles and take them back home. In this case, back home to London. This was a trip I did with Ollie. And as Ollie explained, each of these bottles has all the fish collected at one locality at a specific time. So once these are broken up into different species, you end up with something like that. And that then gets incorporated into the collection. Now, the only area of my research I'm going to want to cover here are mastacembalids, because they also lead to a larger problem. And that's the head of a Macrognathus aral. And mastacembalids are peculiar in that they have this very long extension of their nasal organ and snout. It's called the proboscis of mastacembalids. And at the very tip there, you can see the nostrils. And it's covered in taste buds and neuromass to help it find prey in the mud, in the gravel, in leaf litter, and that's how they forage. Mastacembalids occur in Africa with one species in the Middle East and a large number of species in Asia. There are currently about 93 species described. And when I collected in Myanmar, and I was always already, as an aquarist, interested in mastacembalids because they have this weird look. When I collected in Myanmar, I came across this fish, quite an interesting little animal with these spots, these ocelli along the dorsal fin. And I took a radiograph, and because of the series of little tooth plates in front that support the proboscis, I was able to see it's definitely a species of the genus Macrognathus. And then, of course, you go into the literature, and there's a revision of the Asian mastacembalids by Sufi, and he had this fish under Macrognathus aculeatus. But as you can see from the list of what is this? Sorry. Of the synonyms, these fishes are quite widely distributed, and there are lots of names available for them. Now there's a more recent paper. So how do I get this? Right, so I had to turn to more recent literature, and this is Robert's revision of the genus Macrognathus, and he looked at aculeatus, and he said that's at least 3 different species, and he is illustrating them here, and he looked for the oldest available names for them, and that's aculeatus at the top, aral in the middle, and then siamensis below. And he said that Macrognathus aral, which is the species that occurs in Myanmar, also occurs in Sri Lanka and most river systems of the Indian subcontinent, but it was described from India. All right. This is, and that's what you need to do when you want to work taxonomically. You go back to the original description. That is the original description of Macrognathus aral from Bloch, and he describes in very few Latin words what this species looked like, and he has an illustration in there. You can see that this fish has four large ocelli in its dorsal fin. Now, fortunately, the type of Macrognathus aural is still preserved in the Berlin Museum, and that's what it looks like. Rather terrible specimen in a sense, but remember, this is more than 200 years old. Some of the scales are gone, and the coloration is almost gone, except a little ocellus here at the back. And here below is what aural really looks like when it's freshly caught in a photo tank. But of course, I had to deal with all available names, and one of them is Macrognathus jamuensis, described by Malhotra and Singh in 1975 from Jammu. And that's the illustration of the holotype that accompanied the paper. And by just comparing it briefly, you can already get an idea that Macrognathus jamuensis is probably the same as Macrognathus aural. So already comparing these two, the relatively poor illustration of the holotype of jamuensis and a live specimen of aural makes you wonder and probably convinces you that this is just a synonym of aural. And when you look at how the authors distinguished aural, it was still called Ichthyotaxonomy by them, from their new species, then you see that it's actually little details, not very significant differences, if any, and also that information was not taken from the original description, but from Day's compilation. So who knows what Day looked at when he put this together? He certainly did not look at the type. There was another name I had to look at, and that's Macrognathus morehensis, because this was described from Manipur, from the Chindwin drainage, and so could potentially occur in Myanmar, and could potentially be the species that I collected. Now, that's again an illustration of one of the types, in this case a paratype, and you can't make out really any significant details that would help you. But again, I did know someone in Manipur who could probably help, and that was Vishwanath. And I contacted him, and he sent me a photo of the holotype, for which I was very grateful, which showed me at least roughly what it looked like. And again, you can see that's a relatively poor specimen, probably picked up from the market. It's depigmented, has lost really the important parts of its coloration. And this is what an actual freshly preserved specimen looks like of this species, Macrognathus morehensis. So this clearly was not the species that I had collected. And I'm going to go on to a little detour now and then come back to Macrognathus aural and the fish I collected in Myanmar, because I think it's necessary to mention this. Now, for some reason, Macrognathus vastassemblitz in general seem to attract a lot of people who publish descriptions, and those descriptions don't really help with taxonomy. They confuse taxonomy because they don't look at types, they don't look at the original literature. They just describe species based on a few specimens they collected. And one of those species is Macrognathus albus from southern India. And when you look at the diagnosis, then it would qualify as what Roberto Reyes called a poor diagnosis. And he, if he was editor, would have probably rejected this paper. But if you look at the original description of Macrognathus güntheri from Day, from this area of India, then you just look at the holotype images and his description, and you can see that the general color is greenish brown. There is a yellowish white streak that passes from just above the eye along the lateral line to the center of the caudal. The soft portion of dorsal fin is dotted with brown and has 14 irregular blotches. The caudal has a black bar. So everything that they mentions for güntheri is actually present in this species, and this supposedly new species. So I'm pretty convinced this is just a synonym of Güntheri. And the authors didn't really bother to check if there were any types. And there are syntypes of Güntheri, and they still have some interesting color pattern, which clearly matches what they report for this supposedly new species, Albus. Now, this brings me to a larger problem, which has been mentioned a couple of times, because look where this species description was published in the International Journal of Pure and Applied Zoology. Well, nowadays, if you read international journal, you can almost be sure that it's not an international journal, but it's a national journal published by one country, where most of the editorial board is from that country and the reviewers are from that country. So you don't really get a good peer review. And that brings me to this point, which is frustrating when it comes to Indian freshwater fishes, the unholy alliance, predatory journals and taxonomic vandals, right? So you know that predatory journals are those that charge you for publishing in them, and they usually have a very, very poor or absent peer review process. And taxonomic vandals are those people that just describe new species without a lot of evidence, avoid peer review, and they seek out these predatory journals and publish in them. So Moore et al. Pointed out that bad taxonomic practice characterized by unscientific decisions and professional misconduct is known as taxonomic vandalism. And it can needlessly complicate conservation efforts and other important areas where taxonomic research plays a role. And self-published journals and books that are not peer-reviewed have facilitated taxonomic vandalism. But of course, taxonomic vandalism is first and foremost massively important for taxonomy itself, because it complicates it unnecessarily so. And this brings me to this individual here, who between 2013 and 2020 described 26 new species. Now, there are people who have been more productive, no doubt about it, but all those species in blue are species from Kerala, because that's where this individual was based or is based. And if you can see, Kerala, certainly India and the Western Ghats are a hotspot for freshwater fish diversity. But this person described 7 different species of puntios from Kerala, 5 different mistus, 3 cystomas, 2 pristolapis, 2 macrognathus, and a few others, a Horabagrus, a Cryptothorax, a Rasbora, a Labeo. So clearly, Kerala being one of the best known areas in India taxonomically in terms of fish diversity, it would be extremely difficult to find this number of new species in these genera. And if you look at the journals themselves, there's one that has an interesting name, the International Journal of Innovative Studies in Aquatic Biology and Fisheries. And you can tell when you go to the websites, all of these are predatory journals. You have to pay to publish in them. And because you pay, no one really checks the content. So most of these species, if not all of them, are not valid, not good species. And if somebody goes through the effort, look at them in detail, will probably discover that. But it's almost like a shotgun approach. You just shoot into the forest and one of your bullets might hit an animal, and maybe 1 or 2 of these are actually valid species, but most of them, I'm sure, are not. And if you remember what effort it took to get rid of one of those names, that was Horabagrus melanosoma, and I got this slide from Nilesh's talk, which he gave on the first day, to show that Horabagrus melanosoma is not a valid species, but a synonym of Horabagrus brachysoma. If you know how much effort went into that, you can kind of get an idea how much effort would be needed to get rid of all these, or restudy these 26 different species and get rid of most of the names. But back to Macrognathus. So I knew what Macrognathus aura looks like. I knew the diagnostic characters, and I was able then to compare that with species I collected in Myanmar. And with time, of course, and more collecting effort, I collected more different Macrognathus species, one actually from India itself. And just a brief look at the color pattern of the dorsal fin convinces, I think, everyone that these are so incredibly different that they can't be the same species. And it turns out, after careful study, where you look at radiographs, do measurements, counts, comparing specimens of Aral with those in Myanmar, and look at more than 130 specimens, do counts, as I said, then you come to the conclusion that all these are new species, and they have been described in various papers over the years. So Macrognathus aral does not occur in Myanmar. It's replaced there by different species. They were all new, and there was even a new species in India. Now, this was all studies, these were all studies on Indian fishes I did because I worked in Myanmar and had to compare. But my actual work on Indian fishes kicked off when this man on the right used this lady in the middle to get into the Natural History Museum, because Rema Devi in the middle was visiting in London, asked me if she could visit the collection, look at some day specimens. And while she was doing that, she got a call, passed the phone to me, and there was Rajat. And he said, well, Rema Devi told me she's in the collection. Do you think I can also come? And I thought, yeah, of course, just come. So that's the three of us. We had a great time. We chatted, and that's how my contact to India and Indian ichthyology was established. And then, of course, the next step was to go into the field, and that's where I met these other two Japs. That's Sibi, Anwar, and on the lower right, that's Nikhil. Now, Sibi and Anwar are ichthyologists. Nikhil is is actually the wrong word. He is definitely a aquarist, fish enthusiast, and has a ornamental fish business. But his main drive is his passion for indigenous fishes. And he's been traveling around Karnataka and the southern parts of India to collect fishes, to just see where they occur. So during that trip, we collected in different places together. And he led us to places that we didn't know about before. And we collected interesting fish species. Among them, new species. So the four on the left were new species just collected during that trip. And in the upper right is this fish that you might recognize again from from one of the previous slides, this Carina tetradon imitator. Because during those collecting efforts in one of the rivers, with Nikhil's help, we found this fish. We know now where it occurs. And it does not occur together with Carina tetradon trodoncoricus, but in a different river system much further north and in a different part of the river. So finally, we were able to settle where Carina tetradon imitator really comes from, where it lives. Now, of course, when you visit people, you get to know their students. And this is a shot of Rajav and his group when I visited in 2017, I think. And that's Hiranya Sudasinghe, Rohan Pethiagoda's student there at the front. So I got to meet him. He came over from Sri Lanka. And that was very important because we are now working on different projects together. So not only have I started collaborations with various of Rajav's students, I'm also working with Hiranya now, who, by the way, is now a PhD student in Switzerland. And Unmesh and Anup visited me in London. And of course, during one of those trips, I met Nilesh and Pradeep, a Nilesh student, who is now a PhD student here in Prague, just a 2 hours drive away from Dresden. Right, so these are some of the new fish species we described, new families even. And since 2011, when I went to India the first time, Rajav, his team, his colleagues, and I have published a total of 19 papers describing more than 10 new species, actually 2 new families, and we clarify the status of another 7 species. That is what I would call a very successful collaboration. Now, the last part of my talk, I'm going to talk about Francis Hamilton and his monograph of gangetic fishes. For all the Indians in the audience, I don't really have to explain much. This is the standard monograph on Indian fishes, the first monograph on a group of fishes in Asia ever published in 1822. It's a text volume and a plate volume. And the text volume has 405 pages, and it covers 272 species. And some of these species, but only 97 of them, are illustrated on 39 plates in black and white, in monochrome. When Hamilton's monograph came out, Georges Cuvier, one of the leading systematists and anatomists back then, said, "This is the finest contribution to ichthyology ever received from distant lands," a very, very high praise. And Hora later said, "There is no work of greater importance than an account of the fishes found in the river Ganges by Francis Hamilton for the student of Indian freshwater fishes." But when we look into the history of this monograph, we come across this little unpublished paragraph that did not make it into the published book, and was only in the volume that went to the printer to publish the monograph, but then was cut out. And it reads, "I have to regret that the number of plates is so small, owing to my having been deprived of the drawings of 144 fishes. These, together with the drawings of 20 quadrupeds, of 241 birds, and of 130 plants, the government of Bengal, at the instigation of the Earl of Moira, took from me by one of those mean exertions of power into which a weak man thrust into high authority is liable to fall." So clearly there were more illustrations, 144 more, that he could not take with him back to London and have them published. And what is more, these original illustrations that he had to leave behind and the original illustrations that he used to illustrate his monograph were actually in color. And I've been able to track down all the colored Hamilton illustrations that are preserved in the UK, put them together, and republish Hamilton with these beautifully executed, beautifully drawn, colored images. And I have to stress here that it was not Hamilton who drew these. It was an Indian artist called Haludar who drew most or probably all of Hamilton's fish drawings. So Hamilton trained him, but his expertise is really incredible. And these are just some examples. That's Chanagachua. This was Monotrade Kutkutia, or Tetradon Kutkutia, or Lyodon Kutkutia, as it is now called. That's Chanagachua, that's Botiadario, Bagarius, Oryctes cosuatus, beautiful. And that's Seesaw, Rhabdophorus. And he also illustrated what would now be called Macrognathus aral. And if you then compare Bloch's original drawing of Aculeatum or aral and the specimen with Hamilton's drawing and then a live specimen, you can see the big difference. Hamilton had them in front of him. He was able to have Haludar draw them from fresh specimens, live specimens probably. And Bloch was only able to work from preserved specimens that were sent to him from far away lands. So that was Hamilton's big advantage. And they are really accurate and true to life. Out of these 272 species that Hamilton included, he considered 259 of them to be new species. The other ones were just described species he included because he encountered them. Out of these 259, 70% are still considered valid species today. One of the few times where I actually got it wrong is something like this, where you have this beautifully colored fish there, described as Trichogaster tuna, and this more drab-looking one with a black longitudinal stripe as Trichogaster sota. But these are actually just the male and female of Trichogaster tuna. But mostly he got it really right. What did puzzle ichthyologists for a long time was this black and white drawing that he included in his monograph that is supposed to show Channa barca. Now people have said there is no such fish in India. This is probably Channa stuarti or something like that. But when you actually see the color drawing and then see the first live images of this fish that were sent around at around 2005, you can appreciate the very, very close similarity between the live specimen and his drawing of Channa barca. And Channa barca has a very restricted distribution, and he collected it near Goa Para, where it inhabits perpendicular banks and holes dug like those of the martin. And still today, Channa barca is a rare fish, and it's only encountered now and then. I said 70% are still considered valid today. Well, additional species that are now considered to be, their names considered to be synonymous, will probably have to be resurrected. One example is this one. The one on the right, Ophiocephalus aurantiacus, is considered a synonym of Ophiocephalus gachua. And you know, if you look at the black and white drawings, that might be a reasonable assumption. But if you look at the color drawings, it's pretty clear that Ophiocephalus aurantiacus is a valid species, and somebody just needs to go and recollect it so it can be studied in more detail. What I was also able to do is produce a map of where Hamilton was when. And I produced a table, which I'm not going to show here. It's in my introduction to the republished Hamilton monograph, where I've listed all the species and where Hamilton encountered them for the first time and had them drawn at that point in time. So that gives us an idea about type locality. So even when he mentions in all the rivers of Bengal, the date and the place where he collected the fish, which is available from his field notes, gives us an idea when the specimen that he illustrated was actually drawn. And we can use that to determine type localities, even for species that have such a wide distribution in his monograph. Now, this is what it looks like. That's Francis Hamilton's fishes of the Ganges in color. And this can be bought. It was published in August 2019. And this is us with the first book in hand celebrating this event. It was published by the Race Society. The two gentlemen on the left and right of me are from the Race Society. They also work at the Natural History Museum. And that's Oliver Crimmen on the right, who really helped me get this project finished. That's almost the end of my talk, but I thought I'd give some general advice. So looking back at my career, these would be important points to keep in mind when you want to start a career in science, especially in taxonomy. Try and get a broad academic education. I was really, really privileged, and I did not know that at the time, that I had this broad academic education. Not every university offers that, I do know, but make an effort and find out what courses different universities offer, and then choose the one that best suits your ideas, what you want to do with the rest of your life as an academic. Be passionate, that is very important, be passionate. Roberto Reyes yesterday said that what makes him get up and get out of bed in the morning is his passion. He can't wait to get to the lab to look at fish. And that's how I've been feeling all my life. I want to get up. I want to look again at this diversity. Fishes I have only been able to study for a bit and haven't fully understood. That's what drives me. And that's what, in the end, needs to drive you to be a successful academic, really. Then find mentors who are also passionate and critical, including being self-critical, and who have a good reputation among their peers. Now, that's important. And I can tell you that passionate mentors can spot passionate students, and they can spot students who only pretend to be passionate. Now, if you meet a passionate student as a mentor, you will go out of your way to help the student. So that's a huge advantage. But of course, if you get this feeling the student is not so passionate, you might not do what you would normally do as a mentor. Develop a critical mind. That is extremely important. You have to be critical about what's been published, critical about the big names in ichthyology. You have to be critical about Hamilton, Day, Hora, Britz, right? You have to be critical. You have to challenge your own views. Show your views to your fellow students. Discuss with them what your project is about. And you, if somebody shows you what they're working on, try to be critical. Try to challenge what they think. We had huge discussions when I was a student, which didn't exactly end up in physical fights, but we would argue for hours about certain things. Very important, stay away from researchers who publish in predatory journals, because they will give you a bad reputation. And they already have one. Just ask around, you will find out who that is. And then spend time abroad, expand your horizon, get out of your comfort zone, challenge yourself. As I said, I was really scared when I went to New York, but it changed me and it helped me. So these would be my main points of advice I could give. And that brings me to the end. Thank you. Thanks to all whoever helped me in getting where I am now. They are too numerous to list, but they know who I mean. So thanks.